Good morning. I'm Sana, and you're listening to Let's Grab Coffee on WYXR 91.7 FM. Every Monday morning, I'm joined by experts from across the country who are investigating our most pressing social issues and common curiosities. Over the next hour, you'll learn about their inspirations, motivations, and of course, what they know about the world around us. So grab that cup of coffee and get ready for a fun and insightful conversation. The leading cause of disability in the United States is neuropsychiatric disorders like autism, bipolar disorder, schizophrenia, and neurodegenerative diseases. While these disorders affect millions of people worldwide, there is still much to be learned about treatments. So, today, to talk more about neuropsychiatric disorders and possible interventions, I'm joined by Dr. Héctor de Jesús-Cortés. Dr. de Jesús-Cortés is a postdoctoral fellow in the Picower Institute for Learning and Memory at the Massachusetts Institute of Technology. As a member of the Bear Lab, he studies the physiology of synaptic plasticity and how experience and pharmacological treatments can impact this process. He is also the director of the Sagrado MIT Neuroscience Pre-College Program, a summer internship and mentoring program designed for high school students in grades 9, 10, and 11 who are interested in pursuing a career in science, technology, engineering, and/or math with a focus in neuroscience. Welcome, Dr. Héctor de Jesús-Cortés. It is such a pleasure to have you here with us this morning.
Thank you, Sana. Thank you for the great introduction. I'm very, very happy to be here with you today.
Yes, I was so excited. We got connected via Twitter. So that's the positive side of social media. We get to meet really awesome researchers and just great people. And once I learned about your research, I was like, I have to have him on the show because this is such an important topic. So millions of people around the world are affected by neuropsychiatric disorders, the leading cause of disability in the US. And something, honestly, I never really thought about a lot, even though, I know people who are, on the autism spectrum or might have some of these other disorders. So it's so great to have you. And I'm wondering if you could just kind of start us out by telling us just a little bit more about like what neuropsychiatric disorders are.
Yeah, so neuropsychiatric disorders are a family of, diseases that really have to do with the brain, and also the spinal cord and other parts of the central nervous system. And importantly, what we know about them, it's very little. We know many of them have names, like you mentioned, autism spectrum disorders. We have Alzheimer's disease, Parkinson's disease, because we see a lot of behavioral changes in people. But the underlying mechanisms of how is it that they, in the brain, at the molecular level behave are very, very in the early stages. And that's the reason why we don't have a lot of treatment for them. So now the biggest problem in neuropsychiatric disorders is that we don't have treatments that go to the root of the problem. We only treat basically the symptoms. So under this umbrella of neuropsychiatric disorders, there are a lot of different diseases, but in reality, every day we can learn and discover new ones.
Yes. Now, before we start talking about some of the possible treatments, I'm wondering, are neuropsychiatric disorders, are they becoming more prevalent? Or are we just becoming better at maybe detecting them and maybe diagnosing them?
That's a great question. I think it's both. So one of the things that characterize some of the neuropsychiatric disorders is age. So the aging is a process that is normal, but as we became really, really good at other organs, protecting other organs, we know more about, nutrition, exercise, people are living longer. And therefore, we are seeing more neuropsychiatric disorders emerge. But additionally, we also have better tools to detect them early or detect them at all, because in the past we couldn't. So yeah, it's a little bit of both. And what we're seeing is that the trend is that it's going to become really, really prevalent. Even though it is right now, it's going to become even more in the future.
Okay, so even more prevalent. So it's really good that researchers like yourself and other folks that you work with are starting to learn more and being able to do more types of, I guess, maybe testing or find potential new treatments. So I want to get into some of that because your research is really amazing. Because when I think about these disorders, again, I think that probably all of us know someone, maybe within our families or just kind of immediate circles, who has one of these disorders. And so to think that you and other researchers might be able to find the root cause, right, versus just the symptoms is really exciting. So in one of your, I guess, studies, I saw that you were finding some potential maybe breakthroughs around Rett syndrome. And I wanted to start there, if that's okay with you, because I was not familiar with Rett syndrome. This genetic neurological disorder that's primarily in girls. And I was really interested to find out that it is not, it's not inherited, but it is just a unique neurological disorder, a type of autism, but that it seems like there's not many treatments, I guess, right now for understanding, what is at the root. So could you tell us a little bit about your research as it applies to Rett syndrome?
Yeah, absolutely. So, like you mentioned, Rett syndrome is a rare genetic neurological and developmental disorder that affects, the way the brain develops, causing usually progressive loss of motor skills and speech. And like you also mentioned, it primarily affects girls because it's a genetic disorder in the sex chromosomes. So most babies with Rett seem to develop normally for the first 6 to 8 months, and then they lose skills they previously had, such as the ability to crawl, walk, communicate, or use their hands. So over time, children with Rett have increasing problems with the use of muscles that control movement, coordination, and communication. And it also can cause seizures and intellectual disability. So, they have abnormal hand movements, such as repetitive rubbing or clapping, and replace purposeful hand use. So there are no cures, like you mentioned, but there are potential treatments. And when I was in graduate school doing my thesis, my laboratory discovered this compound, this molecule that we named P7C3. And that compound we knew was able to protect the cells of the brain or neurons from dying. And that's a very, very important discovery because, as you may know, all neurodegenerative disorders are, at the end of the day, end up with cells dying. So if we have a compound that can, help with that or prevent cell death, then that would be a breakthrough. And during my PhD, I was tasked to test this compound in different models, and one of them, animal models, and one of them was a Rett syndrome mouse model, which we had, genetically modified. Other groups have done that, and I had it in the lab. And I would then inject with this P7C3 every day, and then see if the brain was developing correctly and what were the things that we could catch in the brain. And importantly, we found that we were able to alleviate many of the culprits of Rett syndrome. One of them is cell death that happens in the brain as they develop using this compound. And also we found that their anxiety-like behaviors, which is prevalent in these mouse models using different behavioral assays, also became better with the treatment of this compound. So it was very, very exciting, and we published it a few years ago. And we hope, to then go into higher animal models like monkeys and humans to try to see if this is a treatment that could be used for Rett and other neurodegenerative disorders.
Wow, that's so exciting. I can only imagine how excited you were as, day after day, you're monitoring the mice and you're kind of monitoring their cells and seeing that, okay, like something is really happening and that they're sustaining these gains over time. I can only imagine how great that must have
Uh,
felt.
yeah, especially, coming from Puerto Rico, I was reading about the discovery of this molecule, P7C3, and I got so excited. I started talking with the researcher that ultimately became my supervisor as a PhD thesis. And I was so excited to do that. And then we also did it with other models like Parkinson's disease, which had really good promise. So yeah, I was super excited every day to go to the lab, and it was very time consuming and it was a lot of hard work. But at the end of the day, we found out something that could be a hope for these parents and also their patients with this disorder.
Absolutely. And you mentioned that P7C3 also has maybe promising outcomes for other types of degeneration like Parkinson's. So how did you kind of figure out that this might be applicable to other disorders as well?
Yeah, so that was my thesis, and what we hypothesized at the time was like, okay, so we have a compound that we know protects cells from dying. So what are the disorders or models that we could use to kind of prove that this is true in a disease state? Because one thing is to protect a cell from dying that would normally die, and that's how the compound was discovered initially. But then what happens when you have a toxicity, when the environment is toxic and you're about to die? Is this compound potent enough to really protect? And that's when we turn to, the one of the first models was Parkinson, because in Parkinson, we could model it by injecting toxins in the brain in specific regions that control motor movement. And therefore, we have a window, because we know when we inject it, to then treat it with, in this case, P7C3, but whatever you want, and then see if it's sufficient to protect. And that's how we did it. We injected a toxin directly in the brain of mice and rats. And then we also injected the animals systemically. So it's like you're taking a pill with the P7C3 and see if it was sufficient to protect against death in that specific brain region that is called substantia nigra in the brain, which also happens to be in humans and the one area that controls voluntary movement. And what we found was astonishing at the time, which is like when we compare those animals that we injected with P7C3 with, their saline controls that had no drug, we saw many more cells in that area after, the toxin have been implicated in there. So when we saw that, we were like, oh my God, this is really good. But protecting cells from dying is not the same as functional protection, meaning that the cell could be there, but it may be sick, or it could not be doing what it's supposed to be doing, which is like control movement. So we also did behavioral paradigms where we had, the rats moving, and when they are injected with the toxin, they start like running around in circles. When you inject them with another drug called amphetamine, they become very hyperactive and they start like switching. It is like very incredible to see those videos, because they start running around a cage. But then the animals that were injected with P7C3 were able to not do that. So behave normally. They do become hyperactive, but they don't start twitching like that. We also did it in other models like worms. Incredibly, worms also have the same type of neurons as humans, some of them. And these type of neurons are called dopaminergic. And again, they control kind of movement. So in worms, we also put the toxin to kill those neurons and also the drug to protect them. And we saw that the cells were protected, and also their behavior of moving, of crawling also was protected. So all in all, we saw that not only working in worms, but also mice, but also rats, which is like one of our dreams, not only, work in one animal model, but in multiple to make sure that it's going to also translate at the end of the day to humans.
Yes, I did not. First, I'm amazed just in general, overall, because this is just such great news, right? And I know that there's still more development that needs to happen, but still, this is really good news. But also the worms. I never thought about worms and their neurons and how they might be similar to ours. So that just kind of threw me off. But I love to see the little worms crawling. So I'm glad to hear that the models also held up with the worms. Now, thinking about, again, I know that there's several more, steps that would have to continue for this research before, possibly seeing if it's treatment that would work in humans as well. But I'm wondering kind of where are you in that research? Are you continuing with this particular line around the P7C3 molecule, and kind of what might be the next steps?
Yeah. So I move on to my postdoc at MIT, and I'm not working with P7C3 anymore, but I'm doing something that would at the end of the day come back to, developing new treatments. And the reason why I moved a little bit is because one of the main characteristics of the brain is that it communicates not only chemically, but also electrically. Mm-hmm. And when I was in grad school, I realized that I didn't understand very well how, neurons communicated electrically. So I really wanted to gain, more knowledge on that area. And the purpose of that is to, when I have my own lab in the future and I have, my students and my postdocs, we can have a research program that could look at the disorders at different levels, electrically, chemically, pharmacologically, meaning creating new drugs or developing new drugs, and then at the end of the day, bring it to the patient. Because many of these disorders, like Alzheimer's disease and including Parkinson's, you can see very early on changes in the brain that are not observed behaviorally. So by the time that you see a patient of Alzheimer's disease, forgetting stuff, typically there's a lot of accumulation of pathology or bad things happening in the brain, but you don't realize because the brain is very, very good at not showing that until the end. So we need to find ways to detect these disorders very early on. We are doing a very good job with autism, for example, but with Alzheimer's, we don't have that. And Parkinson, we're getting even a little bit better, but not great. So electrically, the brain communicates, sending signals, and those signals become damaged very early on in disorders. And I wanted to, in my lab, use electrophysiology, which is the field that studies electricity in the brain, to detect disorders very early on, so I can then treat them as soon as I can with P7C3, for example, or other new treatments. Because one of the things that frustrated me when I was in grad school was this idea of like I had to have genetic animals or I had to inject the toxin. But what happens if I don't know if an animal is okay or not? Can I detect it without genetics and without me putting a toxin? And one of the ways to do that is electrophysiology. So now I do experiments where I measure the electrical activity of animal models, including mice, to look at the memory and how learning and memory happens in the brain of a mouse, to then use those rules of learning. When they disrupt, then I can treat with specific drugs that enhance that process to come back and then work with the things that we've talked about earlier.
I am so happy that you brought that up, because as a person who, just a lay person who doesn't know, all this information, right, about the brain, I think, of course, with the brain, there would be multiple layers or levels that we have to kind of understand what's happening, but also intervene at different stages. So that definitely makes sense. So I love that you're getting kind of this very comprehensive approach to how might we again, not just treat kind of the symptoms or after we, see some of these symptoms, but how can we kind of from the beginning or in the earliest stages make some sort of intervention. So I love that. Let's take a quick break. You're listening to Let's Grab Coffee on WYXR 91.7 FM. I'm Sana, and I'm here with Dr. Héctor de Jesús-Cortés, and we're talking all about the brain, and in particular, neuropsychiatric disorders and possible interventions. Now, in the first segment or before the break, Héctor, you gave us a lot of really great information around some of the early work that you've done around P7C3 molecule and how it might aid in kind of our treatment of neurological disorders or neurodegenerative disorders. And I'm also thinking about some of your, I guess, earlier work as well. Again, thinking about some different type of neurological or neuropsychiatric disorders. And I'm thinking about your research on around maybe schizophrenia, bipolar disorder, and thinking in particular around, and then you can tell me if I'm saying this wrong, around the CAV 1.2 interventions that you did. So could you tell us a little bit about that?
Yeah, so CAV 1.2 is a receptor that is found in neurons. So again, going back for all your listeners, neurons are the cells of the brain that communicate electrically and also chemically. And in the way they communicate is by having these receptors that are just like little channels in the neurons that allow for ions to go in and out. And then those ions create enough energy for things to move around. And when you move things around, that's how neurons communicate. So in particular, I study a type of receptor in the brain called CAV 1.2. It's an L-type calcium channel. So it allows ions of calcium to get inside of the cell. And they are very, very important for communication. Again, calcium is a positive ion, and positive ions getting inside of the cell are super important for communication. So there's this mutation that makes this channel doesn't work correctly. So basically, we were trying to understand what happens when this channel is not working correctly, which are found in some patients with autism spectrum disorders. So, the hypothesis is, okay, let's study this channel. The idea is that the physiology of the channel is going to be not working correctly, but in my case, I was tasked to see if having this mutation in this channel causes cell death. So again, going back to my earlier work of neurodegeneration or cell death, I wanted to see if this mutation causes that. And at the end of the day, that's what happened. In a particular brain region called the hippocampus, which is very, very important for learning and memory, we found that other people have found that you have stem cells there that create new cells every day. So in the brain, different than in your skin and other organs who can regenerate normally, the brain has specific hubs that it can regenerate, but not everywhere. So let's say you have a car accident or something, and you have a problem in one part of the brain, cells are made somewhere else and they have to migrate there. So it takes some time. So those hubs create new cells every day. And what we found is that the rate of making those cells is the same, but then in animals with the CAV 1.2 mutation versus non-wild type animals, but then they die faster if you have the mutation. By dying faster, what happens is, of course, then you don't have enough cells to replace others or to have normal development, and that causes anxiety-like behaviors in this particular model of a mouse model of autism spectrum disorders. So what we did is going back to P7C3 is okay, we saw this phenotype, this thing that happened in the brain. Let's see if we can stop it. So we started injecting P7C3 very early on, and we found that we were in fact able to protect those neurons from dying faster, and also the anxiety-like behaviors went away or got alleviated, basically. So that was very, very exciting as well, because for these type of disorders, also they are in children, and the type of anxiety pills that we have are not very safe for children. They can change the brain development. So having a new way of treatment of anxiety for children that have autism is very, very important for the field.
Absolutely. Because I'm thinking kind of what you said earlier, if we're just treating the symptoms and then depending upon how we're treating those symptoms, it may actually lead to further complications later on in life. So if we can kind of understand maybe again, the root causes or some of those underlying mechanisms, then we can have better interventions that don't possibly lead to further negative kind of consequences in the future. So this is very exciting. Now, I know you mentioned that you are kind of taking a different approach to some of your current research. And I'm wondering if there are any projects that you're currently working on that you can kind of share with us what you're working on, and maybe even any new exciting findings that you've had so far.
Yeah, so like I mentioned before, I'm studying now the rules of electricity in the brain of animals. And that field, it's called electrophysiology. And synaptic plasticity, which is what I study, means the capacity of the neurons to strengthen or loosen or weaken connections in the brain. So you can think about it as a yin yang. You are not gonna remember everything. Your brain has to conserve energy. So there's some memories that are gonna become really good, especially if you practice them a lot. That's why you study a lot for exams and things like that. But there are some that the brain are just gonna, weaken and then turn over. So those rules of how that happen usually happen at the neuronal level with electrical properties that the neurons that connect really well to each other electrically, meaning like when this one want to connect to the other one, send signals and the other one responds really, really good. It's like when you have a neighbor and you have it really, really close, you hear everything. But if it's a little bit further, you're not going to hear it that well. So to create good connections, you have to have proximity, like structural proximity, but also the biochemical or the things, the proteins that are inside of the neurons have to be well organized. So what I'm doing is I'm implanting electrodes in the visual cortex of mice. So the visual cortex is found in the back of the brain in what is called the occipital lobe. And what I do is I do a neurosurgery where I implant this tiny wire in the visual cortex. So I can have access to the activity of the eyes. So it's a visual cortex, it processes all the activity that we receive from the eyes or the stimulus that we receive from the eyes. And then after the surgery is done and the animal have recovered, I can show him or the animal a movie or something in a screen, and I can have access to the activity in the brain. Wow. So now I have that activity, but how do I know that the animal is learning something? So to understand the rules of learning, what I do is I show a specific pattern in a monitor. And then I show it and I have a specific output or signal that I get. And then I come the next day and I show the exact same pattern and I see the activity. And then I come multiple days later and I can do that. And I can do it to either eye. I can do it with both eyes, or I can do it with one eye. And then what the lab has found in the past, not me, was that when you keep showing a familiar stimuli, so something that he has seen before, the activity grows larger every day until it plateaus. So depending on the age of the animal, if it's super young, it learns super fast, like humans. And then if it's an older animal, it learns a little bit slower, but we all learn.
Mhm.
Now, at the end of the experiment, we also introduce a novel stimuli. So we change the pattern, and what we see is that the activity goes down to what it was in the first day. So the first day, the pattern was a novel one, a new one. So it learns specifically that pattern that you have created. And then what I'm studying is what's happening in the brain at the molecular level, at the electrophysiological level, at different levels that I have studied in the past to understand how that grows larger. Why is it that it's specific to that angle or that pattern? And we can use also drugs to stimulate that to go one way or the other. So in particular, there's another experiment that I do that resembles a disorder of the brain called amblyopia, or commonly known as lazy eye. So you've seen these little kids with a little patch in one of their eyes.
Yeah.
And that's because usually in development, their eyes are not aligned correctly, or they have a cataract, or there are different ways where the eyes, both eyes, are not working properly. If it's catched earlier by an ophthalmologist or your pediatrician, they can do a surgery and put the eyes, align them. But since, it grew that it was not correctly aligned, what happens is that the brain created a really good connection with one eye, but not the other. So the one eye becomes lazy, and the other one is what we call the fellow eye, or the good eye. So what we can do is in humans, is patch or close basically the good eye or the fellow eye to induce plasticity or strengthening of the connections to the brain of the amblyopic eye. And that works really, really nice if you catch it earlier, before the 11 years old. But it doesn't work really that well when you grow older, or if there's no compliance, if the kid gets bullied at school or something and doesn't wear the patch, then it doesn't work really well. So we can do that in animals. We can close the eye of an animal for a specific period of time and cause amblyopia. So if I take a very young animal, 26 days old, and I close one eye for a week or 2 weeks or 3 weeks, depending on the experiment, when I open it, what I found is that the connections to the brain become weakened. So the previous example, we were getting better activity with familiarity. Now we're getting less activity because we closed it, and the brain is like, I don't need these connections. I'm getting a lot of noise, because through the parpad, you do get some ions through the eyelid, but you're not getting enough. So the information is not very good for the brain. It weakens those connections. And then what I'm trying to do is come up with treatments to kind of reboot or bring back those connections back to normal, because we know that we can do that. And in the lab, a particular postdoc before me found that if you use a toxin called tetrodotoxin, or TTX, which is found in pufferfish, you can essentially make the mouse blind for a little bit of time. So you inject it in both eyes or the good eye. And that, like, imagine your computer, when it's giving you a little bit of problem, you restart it. So you turn it off and back on. It's the same thing. So now by using this toxin, the system has to shut down for a little bit. And when it comes back on, now, even though this one didn't receive a lot of connection because it's amblyopic, it has the capacity and the opportunity to go back to what it was before in the system. So what I'm trying to do is to understand why that happens at the molecular level. What are the biochemistry? What are those channels that I talked about earlier that are important to change calcium, and that we then can use other compounds. We don't have to make you blind, but we can use the same rules of the blindness to reboot the system and make it better. So those are the two, things that I'm doing in the lab right now, and I have really good success with a few of them. I cannot talk about the specific compounds, but I do want to say that there's really, really interesting things happening in the lab that we're hopefully wanting to publish this year.
Wow, that is so exciting. I mean, I'm just really blown away by all of it. And just thinking about one, how amazing is our brain to be able to make, kind of all these choices and modifications in order for us to kind of work, or for the brain to work most optimally. Right. And I think that's just so cool. But the other promising, of course, the promising thing about your research is that when we're thinking about conditions like the lazy eye, or just other kind of connections in our brain, there's ways for us to kind of get rewired, so that we can have kind of the most optimal, I guess, output or functioning in our body. So that's really, really exciting. I think it gives me a lot of hope too, because we often think, just kind of in general, like, oh, this is just how I am, or this is just how I think, or this is just, whatever these kind of patterns and routines are that we've created in our brains. But there's always ways for us to kind of reroute, so that we can
Yeah.
maybe see clearly or think clearly. And so that, I think, is something to be hopeful for. So I have to be on the lookout for maybe some exciting findings and new breakthroughs that you and your lab have this year. That's great.
No, absolutely. And like you mentioned, that give us a lot of hope, and also it tells you a little bit of the complexity of the brain and why we're a little bit behind from other organs, from developing drugs. It's just like we're working with so many different levels of complexity that it makes it really, really hard, because, you can fix the electrical part, but then there's the biochemical part. You can feel the behavior, but then the genetics are there. But what we're really understanding is like many of the things of the brain, like in the past, you would thought like, oh, depression is something of the, psychology related, nothing to do with the brain. The person is just weaker or something. Now we're understanding that there's so many chemicals in the brain, so many imbalances, that in reality it is a disorder, and it's something that needs to be treated with with the same respect than other disorders, exactly the same as Alzheimer's, Parkinson, because we need to understand it really well. And, if it needs treatment, pharmacological treatment, use it. And also, therapy and the other tools that psychologists have developed. So it has opened up a lot of our minds to understand the complexity and to be really empathetic with a lot of the different disorders that in the past were not that prevalent.
Yes, I'm so glad that you mentioned that, especially giving that example of depression, because I think it has been very stigmatized. And like you said, some at some points in time, or even some folks might think, oh, you can just feel your way out of it, or like if you just maybe power positive thinking, that it could be okay.
Mm-hmm.
But as you mentioned, no, when we're thinking about the brain, it is so complex. There are multiple different levels and things happening that are impacting something like depression. And like you said, to be able to treat it with the respect it deserves, but also with the empathy that people deserve as we're thinking
Mm-hmm.
about disorders. And again, treatment being, maybe a couple steps behind, right? Because the brain is so complex.
Yeah, no, absolutely. But one thing that we do know that help everything in the brain, it's the circulation. So the one thing that we have in the brain, it's the same like other organs. We have blood that it's supplying the brain with a lot of oxygen, and because the brain is one of the highest consuming energy places in the whole body. So having a great nutrition, exercising, and trying to minimize stress are things that we can do every single day, choices that we can do every single day to minimize the impact later on of these disorders. Because, the genetics you cannot change, but what we have known now, it's like genetics are just a script. What happens is who's gonna read the script? It's gonna be Jennifer Lopez or Brad Pitt or someone that it's just a bad person. So you choose who is gonna read the script, and the decisions that we make every single day with those things that I mentioned are crucial and are things that we all can do right now And it's going to make our lives, a researcher's life, much easier, because then we're going to, there's some genetics that you cannot beat. For example, my wife specifically studies a family in Colombia that has a mutation that predisposes patients 99.9% to have Alzheimer's.
Wow.
So that's it. They cannot change anything in their diet or anything. So those are, we have to study those. But the majority of Alzheimer patients are not like that. They don't have that mutation. The what we call familial Alzheimer's are less than 10% in the world. So the others are a combination of many of our choices that are done every day and things that we don't understand in the genetics and other stuff. So there's a hope on that side as well. If we understand the power of nutrition, exercise, and stress, those things are really, really important that people should take care every single day.
Wow. Thank you so much for sharing that. And I love what you said, that the genetics are just a script, right? And we get some input on who's going to read the script. So that is great news. Well, let's take another quick break. You're listening to Let's Grab Coffee on WYXR 91.7 FM. This is Let's Grab Coffee on WYXR 91.7 FM. I'm Sana, and I'm here with Dr. Héctor de Jesús-Cortés. We've been talking about neuropsychiatric disorders, possible interventions, and a lot of the great research that he is doing. And, one thing that I haven't gotten to ask you yet is, how did you become interested in this line of research? What led you kind of down this road?
Oh, wow. This is a great question. Well, growing up, I really wanted to become a baseball player. And, growing up in Puerto Rico, I just loved baseball. And it wasn't until high school that I had to do a science fair. And my dad at the time was drinking an extract from a plant called Cundiamor, or Mormodica charantia, scientific name. And he would boil the plant and take the extract for his type 2 diabetes. And I always wonder, I was very, very curious of what is it in that extract that make, the sugar levels go down instead of a pill. And I did a science fair where I went to a chemistry lab to separate the ingredients of that plant and then test them individually to see if one of them was the active one or was the combination of all of them. And that project actually made me realize that I love going to the lab, that I love, working with different experiments, talking with other researchers, doing the literature review, and I just got hooked. So I, at the same time, got injured in my triceps, so I couldn't play baseball anymore, and I had to decide what to do. So I went to do my bachelor's degree in the University of Puerto Rico in molecular and cell biology. And the idea was to use, plant medicine for humans in the future. But then I had the opportunity to be in a neuroscience lab, and I just got hooked again, but more specifically towards the brain, because I realized what we've been talking, there's not a lot of that is known about the brain. All of these disorders are devastating, not only for the patient, but for the caregivers, the family. We all know people that have them, and yet there's not much we can do. And I was like, I want to be part of the solution. I applied some of the discipline and the hard work that it's found in sports. and the one that I wanted to become an MLB player. So I was like, okay, I'm going to go all in on this. And I was really, really successful in that lab. At the time we were studying tauopathies, which is a family that Alzheimer's is part of at the mouse model. And then I did grad school in UT Southwestern, Dallas, Texas, with a psychiatrist who introduced me to T7C3 that I have talked about earlier. And then now I'm at Mark Baer's lab at MIT, where I'm trying to, round up my career so I can open up my lab and finish what I started, which is creating new treatments for patients. But now instead of plants, I want to do it with whatever is necessary. If it's a plant, that's fine. If it's a drug, that's fine. And if it's digital therapeutics, which is something that it's now used a lot, which is the power of like VR, for example, virtual reality. You can use the eyes and put specific stimuli, like I mentioned for my mice, and change the plasticity or the strengthening of the brain. So whatever is necessary to treat patients with neurodegenerative disorders.
Wow. Okay. So there's so much there. Well, first, again, I just love that you are just expanding your toolkit, right? So that you will actually be able to provide kind of the best treatment, most effective or comprehensive treatment possible versus just kind of looking at these disorders from one way. So that's awesome. But then I'm really blown away by, you're like, oh, so I was in high school and I just went to this lab and I'm running all these tests on this plant to see which, part of the chemical compound is really the healing properties, right? Or regulating the sugar. And I'm like, wow, that's so awesome to be able to do that in high school, because again, it sounds very complicated to me. And very like, wow, this is like next level. This is so awesome. So first, let me ask you, were you able to figure out what, I guess, component of the plant that was actually contributing to regulating the blood sugar?
So yes, but it wasn't a particular ingredient. It was the combination of all of them. So it was a synergistic properties of all of them combined together were able to have the hypoglycemic effect, which is, regulating sugar levels. And then we won a prize of the science fair in Puerto Rico, and it was just an amazing experience. Yeah.
That's so cool. And I'm wondering, how did your dad feel about kind of your interest in science, and also kind of, I guess, your interest in particularly what he was doing, right? Every day having to, regulate his blood sugar.
It's I think to this day, my parents are like, we didn't even know you were going to turn this way. Like parents would be like, yeah, you're a genius, you're very intelligent. But to be honest with you, wasn't the best student all the time academically. I did have good grades, but they were not, I wasn't at the top of my class, always wanting to play baseball or go out and party with my friends. So they never thought that I was going to become a scientist, and they're not scientists either. But now they're like, oh my gosh, that little experiment that you did that time that we had to take you so I would go to the lab every afternoon after school. I would go to this lab and spend, 2 or 3 hours there every single day. So they had to take me. I didn't have a car. So, they're now realizing that little, effort, or a lot of effort at the time, turned out to be life-changing for me.
Mm-hmm. Wow, that's so cool though. That is such a great kind of introduction and story, right? Foundation to your interest. That is really cool. And I also was really, really liked how you made that correlation between the discipline that you had for baseball and using kind of those same strategies as you're thinking about, okay, well, now I'm interested in this, and how can I apply that same approach to this, program of study?
Yeah. I believe in translatable skills, and something that we now have because of internet and the power of globalization is we have access to whatever we want. So you don't have to become particularly good at something. I became really good at solving problems, at getting super interested in something and going really deep and solving it. So I always encourage my mentees to find their passion, but also be open-minded to things that they didn't even know that could be possible, and try it and see if they like it.
Yes, yes. And I'm also thinking about, the teachers that you had and that access, right? Being able to go to a lab in high school and run these experiments, and, having, a teacher who could, assist you, and also having the materials, right? The actual lab itself. And I'm wondering, how much is that early experience connected to the pre-college program that you're a director of?
Yeah, it's absolutely everything, because I have realized that when you go to college, you go and you start your career, but how do you know what you like? In high school, at least in Puerto Rico, my experience is that you get the same classes in most schools, but you don't realize what research is all about, and understand what a lab environment is. And in my opinion, that early experience that I had created such an impact in me, and I want to give that to other students. So my wife and I are the founders and co-directors of what you mentioned. It's called the Sagrado MIT Neuroscience Pre-College Program. And the idea is, like you mentioned, giving resources at a very early age when they're in 9th, 10th, or 11th grade, when they don't know what they want most of the time, and just exposing them to what STEM is all about, specifically the research part of STEM, because they're taking classes in science. And also, neuroscience. Neuroscience is because we are both neuroscientists, and we focused on that for this is going to be the 3rd year. But the idea is to show these students with seminars and panels, so career development panels, what is possible for them in the future. I didn't know that what was a PhD when I was in high school. I didn't even know when I was the first 2 years of college. So it wasn't until I got in a lab and I realized that. So learning that very early on is, I think, super helpful. Just have it in your mirror. What is science communication? What is science policy? What are the different routes that you can go? Because nowadays you go to college and it's not like before, like that would definitely give you a job. In biology, it's not like that anymore. In reality, there's not many things that you can do with a biology degree. You have to probably go to grad school for other things to become specialized. So knowing that from the get-go, I would tell you, okay, I should go that route or not go that route if you don't feel that's something you want to do. And, exposure is super important. Secondly, like you mentioned, if you don't have the resources, it's almost impossible. So this program, since it's free, it could reach everyone in Puerto Rico for now. So in the first year, we had more than 500 applications, and from all over the island, which is really, really good, because they're like anywhere, the city, the capital is San Juan, and that's where the best schools are and like the best resources for the most part. So we were able to capture a lot of students that were like, we didn't even know this existed. And thirdly, there's a lot of things about college that people don't know. Like my own sister, for example, didn't know that, if you go to places like MIT and Harvard, there's a lot of financial aid. So the first thing parents think when going to college for their kids is like, it's going to be too expensive. I'm not going to be able to pay for it. That's it. But in reality, if you don't have a lot of money, these are need-based places that most of the time give you fellowships or grants that allow you to study. But what you get out of it, if you have a degree from these prestigious universities, it's a very, very good head start in life that would, be really good for you. So it's just exposing these kids to things that in reality, when they get exposed in college, it could be too late, or it could be something that they're like, no, I'm not going to do that. I already have a degree in this or that. So before they have all that, expose them, give them resources. And at the end, we also connect them with a mentor that would give them, every month we'll meet with them to see what they want to do, how they can help. And they're at the master's, PhD, or professor level to give continuity to these type of programs. Because, if you're in a school with no resources, you go through this experience, you go back, and then you don't have resources anymore, that's it. But by giving them continuation with these mentors, we're able to give them, the social mobility that we want to try to create with this type of program.
Wow, that is so awesome. I mean, everything you said, I think is so important, early exposure to this type of field for students, but also those kind of networks of support as folks are navigating some of these, educational systems that might seem very daunting. So knowing, again, bring that hope back in that, hey, this is something that you're good at, that there's support for, also that you don't have to, be so stressed about the cost, right? Because, people, we don't know what we don't know. So even just having that exposure, I think is extremely helpful. Well, Dr. De Jesús-Cortés, it has been such a pleasure to have you here with us this morning. I've learned so much. And again, like I said, I'm looking forward to seeing, research that you and your lab have, coming out in this area as well. So thank you.
Oh, thank you, Sana, for the invitation. And please, for all your listeners, you can email me at any time to, if you want me to explain something further, wanted me to understand more about what I do, or if you're in the MIT area and want to just, grab coffee like this podcast, we could also do that. So my email is dejesus, DEJESUS at MIT.edu. So please feel free to contact me anytime. I'm happy to talk about this further, and really, really happy that you invited me. Thank you very much.
Absolutely. Thank you. Thank you again to Dr. Héctor de Jesús-Cortés. What an enlightening conversation. I learned so much. And most of all, I'm just really hopeful for what we might see in the future as far as treatments for these different neurodegenerative disorders and neuropsychiatric disorders. So that is very much exciting news. And just to reiterate, if you want to get in touch with Dr. de Jesús-Cortés, he offered his email. So you can feel free to email him your questions, and his email I'll include in the show notes as well. So for today's positive note, I just want to reiterate something that Dr. de Jesús-Cortés said, which is genetics are just a script, but who is going to read that script? And I love that, because sometimes I think we can feel like, maybe we know something about our genetic history or genetic disposition, but it's just a script, and we do have some input into that ourselves. So thinking about nutrition, thinking about exercise, and thinking about ways that we can lower that negative stress. Well, this has been Let's Grab Coffee on 91.7 FM WYXR every Monday morning. I'm here having great conversations with researchers and experts from across the country. So I hope you will join me again. If you missed the earlier parts of this episode, don't worry, you can hear the replay on WYXR.org on the show page for Let's Grab Coffee, or you could always subscribe in the podcast format on your favorite podcast streaming app. I cannot wait until you and I get to be back here together next Monday.